Welcome again. As you know, research on ALS is continually advancing, and at Everything ALS, we are dedicated to uncovering new findings, enabling early detection, and working towards a cure for ALS. You can assist us by participating in our speech study. It's pretty basic and simple. The goal of the speech study is to enhance the diagnosis and prognosis of ALS. We are looking for participants to provide 5 speech recordings, each lasting 15 minutes, once a month, totaling 75 minutes over 5 months. We invite both healthy individuals and those diagnosed or suspected of having ALS to join us. To enroll, please visit us at everythingals.org/speech project and complete the form. That link will be in our chat, so you can copy it from there. And additionally, we are actively recruiting for another study, the asymptomatic gene carrier study. If you are a gene carrier who is asymptomatic and interested in remotely monitoring your speech, cognition, and motor skills, please visit everythingals.org/asymptomatic-study and complete the form. Thank you for joining a team that's going to make a difference with you helping. Thank you very much. And with that, we'll turn it over to you, Christian.
All right. Well, thanks, everybody. And thanks for joining. We've got a good-sized group already tonight. And we are really excited to have Dr. Hande Ozdinler joining us today. And her topic is called Brain Matters in ALS. And as understated as the topic is, you're going to be really, I think everyone's going to really dive deep with her into the role of upper motor neurons in ALS and understand much more about the research that's being done and her groundbreaking work. And I think as I met her and was immediately impacted by her energy, really inspired by the work she's doing. So without further ado, I'm going to introduce Dr. Ozdinler. Dr. Ozdinler is the Associate Professor at the Department of Neurology in Northwestern University at the Feinberg School of Medicine. And she's also a faculty member at the Les Turner ALS Center, Chemistry of Life Processes Institute, and the Masulim Cognitive Neurology and Alzheimer's Disease Center, and the Robert H. Lurie Comprehensive Cancer Center. The Ozdinler Lab is interested in understanding the molecular and cellular basis of neuronal vulnerability with a focus on the upper motor neurons, or UMNs, that degenerate in ALS, HSP, and PLS. The Ozdinler Lab generated and characterized the first reporter line for upper motor neurons and developed a novel drug discovery kind of verification platform that incorporates improved UMN health as a readout. Together with Dr. Silverman, they identified NU9, the first compound that improves the health of UMNs that are diseased due to misfolded SOD1 toxicity and TDP43 pathology. Dr. Ozdinler is currently working towards understanding the mode of action of NU9 and for the identification of pharmacokinetic biomarkers and that inform about the timing and extent of UMN loss. The lab also works to develop personalized medicine strategies with the use of novel omic approaches and gene delivery strategies so that there will be no patient left behind when it comes to rare and devastating diseases like ALS. Without further ado, Dr. Ozdinler, the audience is yours.
Thank you so much. What a great introduction, and thank you so much for the invitation. I'm really happy and thrilled to be with you tonight. And yes, we have so much to cover, and I think the brain component of ALS requires more attention, and hopefully by the end of this talk, we will all realize that the brain is important, and especially within the context of ALS. So let me start by giving an outline. And I will be, within this an hour, we will be together. Maybe I will talk about the role of the cortical component of motor neuron circuitry. Why the brain plays an important role in this motor neuron circuitry. And I will define the motor neuron circuitry. And why is it challenging to study the brain component? Why people have not really delved into the brain. There must be some challenges, right? So let's name those challenges, and then by the fact that we understand those challenges, we can actually begin to overcome them. And then how can we incorporate this cortical component of ALS into drug discovery efforts. And I will tell a little bit about our story. But then what are the roadblocks, right? And why cannot we move faster into developing drugs, especially for the cortical component? And I will touch upon biomarkers and, how they play a role in the drug discovery efforts. So let's talk about the motor neuron circuitry. What do we mean by motor neuron circuitry? So, brain is complex, as we know, and this was a drawing made by Ramon y Cajal more than 100 years ago. And he was one of the first scientists realizing that not all neurons in the brain are look alike. They are different. They look different, okay? So that he realized the complexity of the neuron or complexity of the brain, and that there were many different neuron types. And then he basically described this large pyramidal neuron population sitting at the deep layers of the brain and sending long apical dendrites towards the top of the brain, all right? And then these are like humongous, huge, big neurons that don't look like anything else. And he also realized that there were other neurons with many branches, arborizations, small cell bodies, maybe some astrocytes, maybe different neurons. And, he made many, many drawings of these large pyramidal neurons. Later on, we found that they were in layer 5 of the brain, deep brain. Now, we call them upper motor neurons. Some people call them cortical spinal projection neurons, cortical spinal motor neurons. There are so many different names for them. The field even cannot pick one name for them, all right? They couldn't even decide what to call them. Everybody has a different name, but we call them cortical spinal motor neuron. So people sometimes don't agree with me because I call them motor neuron. They say they're not motor neuron because they don't have connections to the muscle and they don't execute muscle function. They must not be motor neuron. I understand, but I want to emphasize that they are within the motor neuron circuitry, and to gain them more respect, I want to call them corticospinal motor neuron. When you say corticospinal, it means they reside in the cortex and they project to the spinal cord. That's why corticospinal. For example, if you have thalamocortical, that means the cell body is in the thalamus and projection is to the cortex. So it's corticospinal, which means cell body is in the cortex, projection towards the spinal cord, and they are within the motor neuron circuitry. Because the motor neuron circuitry, in the most simplistic term, is that these are the corticospinal neurons in the brain. They project all the way to the spinal cord, but in the spinal cord, there are also other motor neurons, which people call lower motor neurons, spinal motor neurons. But these neurons are extremely important because they project to the muscle and they are the ones which contract the muscle, okay? So then I would imagine that these are like the neurons that give the order to these spinal motor neurons, and in turn, spinal motor neurons have the executive function. But they're like the CEO of the company, they initiate movement, they modulate movement, like they are the brain of the movement that happens. And imagine this, like you are about to reach this glass, right? And we reach for a glass, we open the door, we dance, we talk, we use our hands. And to us, it's such a normal or easy thing. But in fact, being able to reach for something, right, and grasp something, you know how many different neurons are involved? It's huge. First, the visual system has an input because you need to see it, right? And also, if you are grasping it, if you are holding it, you also have input from the sensory nervous system because you don't want to crush it. And you need to be just the right touch. And then you have to have the cognitive function because let's say if it is hot, you move it further. So you need to assess what is going on in your environment, right? And then different neuron populations have input to the corticospinal motor neurons, hundreds, maybe thousands of them. They speak to them and they say, do this, activate this, and then they integrate all that information and they convey it as one signal to the spinal cord, to the spinal motor neurons, and then it leads to very precise movement. When I say very precise movement, I really mean it, because if you watch your body and say, for example, when you move just one hand, the other hand doesn't move. When you move one feet, the other feet doesn't move. You can move individual finger, like, we can make very precise movements. When the order goes for movement, not like we don't start moving everything. Our movement is very precise and very well orchestrated and organized. So that's why the input going from the brain to the spinal cord is very well controlled. And that's why the upper motor neurons are extremely important for this, because they initiate movement, they modulate movement. Any voluntary movement that we make, it is actually the upper motor neurons that we have to thank. And we should be thankful for them for being able to initiate this. And look how beautiful they are. And they're almost like a sequoia tree, like in the forest, right? Their cell bodies maybe 5-10 times larger. They have a very long, thick apical dendrite going all the way to the top layers of the cortex. And that's exactly where they integrate the information from callosal projection neurons, from nearby other neurons, their other projection neurons into neurons. All that information is integrated. And then with their axon, they send the information all the way to the spinal cord. So we have very few of them in our brain. If you even look in the motor column, like in the motor cortex, their population, percent distribution of their population is about 1% or maybe less. Imagine that. So even where they are located in the motor cortex, they're only 1%. In the whole brain, they're 0.00001, like they're almost non-existent. So that's why when you take the whole brain and you do all these protein studies, or this study, cell study, they're in the background. They are so small, so few, we can't really understand them or study them in a cell type specific manner. And that's why, they're usually lost in translation. But they are one of the most beautiful neurons in the brain. And these are the upper motor neurons or cortical spinal motor neurons in human. And look again how beautiful, how large they are with their apical dendrites, and sometimes you can even see the spines. But in in ALS patients, in ALS FTD patients, you see them degenerating. You can even see like they lose their apical dendrites. Look at their cell bodies has shrunk, and the apical dendrites are filled with vacuoles. They are falling apart. And what makes them unhappy is the most important question, because once we make them happy again, I think that we would be able to develop effective treatment strategies for ALS. But unfortunately, the field did not really appreciate their importance. And there were so many people saying like, who cares about upper motor neurons? They're very few in numbers. They don't contribute to the disease. They're just very limited, and they will die eventually, because people were thinking that they're going to die because the spinal motor neurons die. And let's not focus on them. Why bother? Patients don't die because they die, and there's no upper motor neuron loss in patients. Why bother? And I've heard all those, but it's very depressing, to live in a time where it's easier to break an atom than a prejudice. Because I think that believing that upper motor neurons don't matter is not factual anymore. But, it's very hard to break that line of thinking. And we cannot really solve our problems with the same thinking that we used when we created them, because some line of thought is actually the source of the problem. So then I think we need to understand that upper motor neurons are an important component of the motor neuron circuitry, and they degenerate early in ALS, which I'm going to show you, and they contribute to disease pathology, and they are important cellular targets for effective treatment strategies. I mean, especially because in ALS, it's the voluntary movement that is affected. And if upper motor neurons are the neurons that are important for the initiation of that movement, why not target them? Why not make them happy? I think that this would lead to long-term effective treatment strategies. And if you look at the upper motor neurons, I remember I told you this apical dendrite degeneration, and you may say, like, why is this so important, right? Why apical dendrite degeneration is so important? So apical dendrites are the sites where all other neurons begin to talk to the upper motor neurons, okay? And neurons speak to each other by synaptic connections. So there are many, many, many synapses along the apical dendrites. And to those synapses, the excitotory neurons come and talk to them. Inhibitory neurons come talk to them. Many different neurons come talk to them. But if you have an apical dendrite, which looks like this, Swiss cheese, there's no way that these neurons will be modulated properly, right? Because then they won't be able to hear or understand what the brain is telling them. And they will be left out of the circuitry. They won't be able to be in the groove and be in the group, and they won't be able to hear what the other neurons are telling them. And they may not be able to start an action potential. And they may not be able to send the message because they can't even receive the message. And we realized that this apical dendrite degeneration is a very, very, very early event in ALS. Even before patients show any clinical symptom, there is apical dendrite degeneration. And this happens in the sporadic ALS cases, familial ALS cases. But if you look at the Alzheimer's disease, it doesn't happen because the upper motor neurons do not degenerate in Alzheimer's disease patients. And then this phenomena, like this apical dendrite degeneration phenomena, is also observed in SOD1 model, TDP model, profilin model, spastin model, whatever disease models that you are looking where you see corticospinal motor neuron degeneration, you see apical dendrite degeneration. And many labs also shown that there is spine loss even before apical dendrite degeneration. So then this profound and early apical dendrite degeneration begins to tell us that the cortical component of ALS is actually affected very early in the disease, that they cannot receive proper input from the cortex. And look, you can definitely see the defect. And we have shown that this happens both in the sporadic and familial cases. So regardless of the mutation, this pathology, this cellular problem is present both in sporadic and familial cases. And again, normally they would look like that. But it's not just the apical dendrite degeneration. There is also axonal degeneration. So axon is the part which projects to the cell, from the cell body all the way to the spinal cord. So they form the corticospinal tract. And it is one of the longest tracts because imagine it goes from the brain all the way to the spinal cord. I'm not very tall, but for tall people, that could be like a meter long corticospinal, the axon tract. It's huge. And those neurons have to maintain this axon till you die. They have to have that established and maintained for a very long time. And we see corticospinal tract degeneration in ALS patients. So imagine this, that a neuron is losing its ability to receive input, and it is losing its ability to convey the input. So how can we say that they are not affected? It's not possible for us to say, they are just fine. They are not just fine. They cannot receive input. They cannot convey input. But then why do people say they are fine? Because the definition of problem is different among different people and different scientists. For example, some of the pathologists, they look at cell loss. Like, let's say they do Nguyen staining, Nissl staining, and they want to see the cell body. If the cell body is there, that they can see the cell, then they say, well, the cell is here. There's no cell loss. If there's no cell loss, then there may not be a major problem, right? Because they see the cell. But then I have the counter argument for this, because just because, the cell occupies space doesn't mean the cell is functional. And sometimes I tell, for example, just because I'm in my office doesn't mean I'm working, right? So the cell body may be there, but we don't know how functional it is. It may be just like, okay, I'm not going to talk to you. Okay, I'm just here. I'm just sitting here. And you may see the cell with new initial staining, but it's not doing anything. So then there may be a functional problem. So the cell biologist may say, all right, the cell is here, but are there any problems with the cell? And if they see the cell, cell body this intact or that, and say, we don't know if there's neurodegeneration, if, for example, problems with mitochondria, with nuclear membrane, with this, with that. But they look inside the cell, which is actually very important. And molecular biologists take even a deeper approach, and they say, what is the molecular basis of this neurodegeneration? At a molecular level, are genes, proteins, protein-protein interactions. And now, at that level, you actually begin to reveal the basis of selective vulnerability. And I think that moving our attention to the molecular biology and cell biology will be very influential in understanding what is really the main problem. Because if you just do Nissl staining or Nguyen staining, you would still see the cell bodies with single dots. And if you count them, you may say, oh, they're pretty much the same. But it's actually not the same. It's because the cells are shrinking, cells are not doing what they're supposed to do. Just seeing the cell body does not tell us that there's nothing wrong with the upper motor neurons. They're actually losing their apical dendrites, they're losing their connections, and they cannot send the signals to the corticospinal tract. So people trying to understand this, the involvement of cortex in ALS, some people say, oh, it's a die-back mechanism, which means that it's, again, die back or die forward. They both think that it is linear, okay? Either going from cortex to the spinal cord, which is die forward. They say brain gets ill or sick or diseased first, and it goes to the corticospinal tract, then the spinal motor neuron, then the neuromuscular junction at the muscle. Okay, so this is die forward. But die back people say, oh, it is going from neuromuscular junction, spinal motor neuron, corticospinal tract, and then the cortex. It's just like linear one way, right? But, the spoiler is that life is not like that. Life doesn't work like that. It's not linear. And it may be that both of these neurons are vulnerable. They are both unhappy. And the motor neuron circuitry is falling apart. It's just like, the London Bridge is falling apart. It's not this leg or that leg. The whole bridge is falling apart. The motor neuron circuitry is falling apart. And I don't think it is linear. It could be very complex. And it's also, we know that other neurons like interneurons, astrocytes, microglia, other neurons that are involved in that circuitry also play a role, right? It's not just these two neuron populations. And most importantly, we call them upper motor neuron, lower motor neuron. It's as if there's this just one neuron population. One lives in the top floor, the other one lives in the bottom floor. Okay? That's not the case. They are born to different neuronal progenitors. Their requirement for survival are different. They express different transcription factors. Their birth and specification development are totally different. So just because we call them upper, lower of motor neuron doesn't make them the same neuron type. Their requirements are totally different. They are distinct and different neuron populations. So that's why I suggest not to be on, picking sides as if they're picking football teams, right? It's not die back, it's not die forward. It's basically the whole motor neuron circuitry is falling apart, and we are responsible of making everyone happy. So if we focus our attention just to spinal motor neurons, how can we build an effective bridge, right? And how can we build an effective and long-term treatment strategy? We have to include the cortical component into the picture as well. And as I was saying before we started, for example, for preclinical studies, there aren't really assays to include the upper motor neuron survival as a readout. And even if you look at the clinical trials now, believe it or not, none of the compounds search or investigate whether their compound of interest also improves the health of diseased upper motor neurons. They don't investigate. And why not? Because those neurons also degenerate in ALS patients. So we advocate for, keeping the motor neurons in the brain or the upper motor neurons in the picture as well. And again, these were many papers showing how much they're involved. And, there are papers after papers showing that corticospinal motor neurons are important. And this is actually one nice review from the Wookiee group, that this cortical hyperexcitation, which occurs 6 months, maybe a year, even before any symptom onset in patients, can even be used as a diagnostic, may have a diagnostic implication. Because the cortical spinal motor neurons, the upper motor neurons, they become hyperexcited very early in the disease, and then they go hypoexcited. Like they say, oh, there's something wrong with me. There's something wrong. And I says, nobody's talking to me anymore. And then they give up. So this hyperexcited time is actually, patients may not even show any clinical symptom, but they are actually having their brain giving the red flag. So now, you remember that people were saying that this is a dieback mechanism and upper motor neurons are going to die anyways. Actually, I want to give a shout out to Caroline Rouleau. So she published a very nice paper in Annals of Neurology 2020. And she used mouse genetics and crossed the FESF null mice with the SOD1. So FESF is a transcription factor that is required for the birth and specification of upper motor neurons. So in the absence of FESF, you may not have upper motor neurons. So she crossed them with the SOD1. So you would imagine that these neurons don't have upper motor neurons. So if the disease went all the way from, neuromuscular junction, spinal cord, cortex, whatever, and then if really cortex didn't matter, nothing would have changed, right? They have upper motor, they don't have upper motor, nothing would have changed. But she realized that the absence of those subcortical was actually protective. So that she basically said, it can't be just dieback mechanism. See, the diseased upper motor neurons contribute to disease pathology. I think that was a very nice paper showing that it cannot be explained by dieback mechanism, that the brain is important. And then Dr. Brown actually wrote a very nice review about this, showing that the upper motor neurons are indeed important, and not only important, they contribute to disease pathology, all right? So the second evidence came actually from our lab, and we used we used two things. First, we have taken this UCHL1 null mice and depleted UCHL1 either in the cortex or in the spinal cord using this Cree-Lux model and using different models where gene expression can be deleted only in the layer 5 motor cortex or in the spinal cord. Then we have also used viruses to investigate their degeneration over time. At the end of the day, we found that when you deplete this UCHL1 gene, only in the spinal cord, right, or only in the brain, right, and if there was this line of thought that upper motor neuron degeneration is a byproduct of spinal motor neuron loss, you would see upper motor neuron loss only when it is depleted in the spinal motor neurons. And that wasn't the case. So degeneration in the spinal motor neuron did not really contribute to the upper motor neuron loss. So then this, the upper motor neuron degeneration was independent of spinal motor neuron degeneration. And that spinal motor neurons and upper motor neurons were indeed very different neuron populations. So then we published this in Nature Gene Therapy, and they actually made it to the cover because we also found that they, meaning these upper motor neurons, were indeed cellular targets for gene therapy. And that's exactly very important, especially for patients with rare mutations, and that there are no drugs for them and the companies are not interested for them, so that we may develop actually gene therapy approaches for patients whose upper motor neurons are affected. And again, we need to have collaboration with the companies who produce the viruses and so forth. But we are building the framework for those studies. And then the third study came from Clive Svensson group, okay? And that's also very interesting work, because imagine that in the SOD1 mice, right, they corrected the SOD1 mutation only in the motor cortex, not in the spinal cord, only in the motor cortex. And guess what happened? When they corrected this misfolded SOD1 only in the cortex, there was improvement in the spinal motor neurons. There was improvement in survival. There was even improvement in the neuromuscular junction. How do you like that? So then, Clive's group begin to suggest that if we develop therapies to the upper motor neurons, we may actually have a good expectation that we may affect the whole motor neuron circuitry. So that's why they actually started clinical trials improving the upper motor neurons. So hopefully by those three examples, I begin to explain why upper motor neurons are important, why they degenerate early, and how we can actually begin to modulate their health. And by making them happier, we can improve the health of the motor neuron circuitry. So that's why it's really important finding a drug or a solution to improve the health of upper motor neurons. So, I don't have much time to go all over everyone's work, but Mark Bellingham has done phenomenal work showing that there's early spine loss, very early, even before cell loss, even before apical dendrite degeneration in the TDP pathology in those upper motor neurons. Catherine Blizzard's work, please follow her work. And she showed that when the TDP mislocalization happens, right? And this leads to upper motor neuron degeneration very early in ALS. And, Bradley Turner, and he has done extensive studies with metabolomics and lipid changes and how these are very early in the disease. So, Wukik's group I mentioned about, and I mentioned many other studies before, even Wukik's group, that laid the foundation for the importance of the upper motor neurons. So hopefully, we all understand that the brain is important. So then who cares about upper motor neurons? They're going to die anyways. Oh my God, when we hear, such arguments, maybe we should say the movement starts in the brain. The upper motor neurons conveys the brain's information to spinal cord targets. And, they are the spokesperson of the brain when it comes to movement. And the upper motor neuron degeneration does not depend on spinal motor neuron loss. And the brain component really contributes to disease pathology. And improving the upper motor neuron health has a huge impact on improving the motor neuron circuitry. And upper motor neurons are indeed cellular targets for building effective treatment strategies. So then first we need to change our mindset and begin to appreciate the importance of upper motor neurons, because if we don't appreciate them, we won't pay attention. But I think it is time that we pay attention to upper motor neurons, especially within the context of ALS. All right, so let's say you all agree with me that upper motor neurons are important. But then what's next? Yes, they're important, but what shall we do, right? How can we develop translational research? And, we are working with mouse models, this model, that model, but where's the translation? Are we just doing experiments with different mouse models and there's no translation to human? Because at the end of the day, our goal is not to cure the mice. Our goal is to cure the human, right? Where is the translation? Is it at the species level? Is it at the cellular level? Like, where is the translation? That is critically important. And then, how can we simplify the complex problem? Especially the brain is so complex, the disease is so complex, it's so heterogeneous. I mean, we are problems of matrices, right? Like, there are so many problems at so many different levels. How can we make it simple? And then Let's say we made it simple. Let's say we have a solution, but then how do we determine success? How do we know that the drug worked? How are we quantified? How will we know that we have a good clinical trial, that we have a good outcome, right? So these are some of the questions that I'm going to delve into now. But first, let me introduce the cell theory. So this cell theory, I think it's very important. It was postulated in 1939 by two scientists. But again, before then, Dr. Hoek actually showed what a cell is using a cork, And so in 1939, they realized that cell is the foundational unit of life, which means every living organism has a cell. Okay? And that the cell, if you look at it here, has different components. The nucleus, the endoplasmic reticulum, the mitochondria, the Golgi, the cell membrane, the nuclear membrane, those are the important elements in the cell. And interestingly, if you look at yeast, okay, yeast is very simple, right? Yeast is a cell. You look at a bacteria, and then you also look at the cells that we have in our bodies, and then the cells that, let's say, the mouse has, the monkeys have, the dogs have, the cats have, like, all living things, right? Let's say mammals, all mammals, they have cells, right? You would be surprised how similar those specialized cells are. Let's say if you look at a kidney cell in a primate, and a kidney cell in a human, a kidney cell in a pig, kidney cell in dog, like that, I can guarantee you, if you were given slides and you are looking under the microscope, you cannot tell which species it is. So at the denominator, if you put cells, or in our case, the neurons, in our case, the upper motor neurons, it is very unlikely that you would be able to distinguish the upper motor neuron of a human, the upper motor neuron of a primate, the upper motor neuron of a mouse, of a dog, of a cat. They are all excitatory large pyramidal neurons. They are in motor cortex. They collect information. They integrate information. They send information. They may have different ion channels different, maybe some transcription factors different. Maybe their axonal path is a bit different. But at a cellular level, at a molecular level, at a gene expression level, at the differentiation, development, birth, specification, they have so much in common. And then that's why we think that the cell is actually the denominator, and the cell speaks, and the cell has immense information. Let me see if I can change the slide. Yeah. So that's why we think that the translation is at the cellular level, which means if we can understand the biology of the upper motor neuron at a cell type specific manner, then we will be able to understand the biology and the pathology of the upper motor neurons in the patients. Because if you look here, for example, here's the mouse and here's the human. They have the same problem. Apical dendrites degenerate. Yeah, mouse has the same problem. Human has the same problem. They have nuclear membrane problems. Yes, mouse has the same problem. Human has the same problem. They have mitochondria problems. Yes, mouse upper motor neuron has the same problem. Human upper motor neuron has the same problem. They have ER problems. Yes, mouse has the same. Human has the same. They have protein aggregation problem. Yes, mouse is the same. Human is the same. So then at a cellular level, the problems that they are faced with are the same. So then to begin to understand those problems at a molecular level, I think that if we focus our attention to the cells, to the neurons, we would be pretty good. And, we published this finding in within the context of TDP pathology, that, postmortem human samples with maybe I should continue. And then we looked at the SOD1 model, and we show the same thing in patients. So then we realize that focusing our attention to the neurons are extremely important. But then how do we do that, right? How do we do that? Brain is so complex, it's so heterogeneous, and that's why we had to generate the reporter line in which the upper motor neurons are fluorescent. And we published this, so I'm not going to go into detail. And the beauty of that is when you put them in culture, the upper motor neurons remain their GFP expression, which allows us to do drug discovery efforts. Okay, so I'm going to come back to that. So now let's talk about the complexity of the disease. Because, patients may have different mutations. Patients may be sporadic, maybe without a mutation, but the underlying cause of the disease for each patient could be different. One of them may have TDP pathology, the other one, they mainly suffer from oxidative stress or lipid dysfunction or cytoarchitectural problems that axon transport is not good, or they have excitatory problems. Like they have many problems, but some problems may be dominant, right? So then here, for example, this is how they look from outside because they have similar clinical manifestations, but this is how they look at a molecular level, so that they may differ in the underlying cause of the disease. So then, when you give a drug without knowing who's who, right, you give them to everyone, one drug, and if this drug is something that works on, I don't know, mitochondria, so there may be a couple people who actually benefit from this, and those may be the one who develop the disease due to mitochondrial defects, and some may not really benefit from it because it's not mitochondria and so forth. But if we were to understand who is who and what is the mechanism that is the reason for that disease, and we select those patients and give them exactly the drug that they need, then we may improve the success rate of clinical trials. So that's why we need to develop assay systems where we understand the underlying cause of the disease and test our drug based on the mechanism, based on the underlying cause. So that's why using different mouse models is very important, because each of these mouse models represent different underlying cause. For example, the SOD1 G93A mouse model was generated based on the mutations detected in the SOD1. So it's oxidative stress, but there are also some problems with mitochondria. But SOD and TDP do not overlap too much. And TDP pathology, there are nuclear membrane defects. There's also problem with mitochondria protein aggregation. And with Alzheimer's, it's mainly in the Golgi and lipid. And in spastin, it's actually a different disease, hereditary spastic paraplegia. So it's in profilin, it's actin cytoskeleton. Different mouse models have, they represent different underlying cause. So then, when you cross them to this reporter line, now you have reporter lines of disease, CSMN, that develop the disease due to different underlying cause. And you know where I'm getting at, because then you can test your drug in different model systems, and you can identify whether your compound of interest would improve the health of upper motor neurons disease by A, disease by B, disease by C, and that would be a very important information. So then, we characterized all these mouse models that we use, and we know the timing and extent of upper motor neuron loss, like in the Alzheimer's mice, in the profilin mice, in the TDP mice, in the SOD1 mice, and so forth. So now we have about 7 different reporter lines of upper motor neurons in our lab, and this is the largest collection in the world. So I beg, all the drug companies, you have a good model, good compound, please work with us so that we can help you identify, we can help you make better educated guesses whether your compound of interest would also improve the health of upper motor neurons that are diseased due to different underlying cause. I think you need that information before you invest $10-20 million moving into clinical trials. So now we received DoD grant to develop a new platform using diseased upper motor neurons so that we can actually add the drug and watch. Because if neurons are happy, they will send a long axon, they will branch, arborize, and if they are not happy, they're going to shrink, they're going to die. So basically, upper motor neurons begin to speak to us. So we learn directly from them whether they like it or not. So now we can identify, we're trying to identify compounds that respond to drug treatment, right? The compounds, and that they have longer axons and branch arborize. And this is actually how we can trace them, how we can quantitatively assess their response to treatment. And that's, a very good way to assess the upper motor neurons. And Chris in our lab has developed this high density multi-electrode arrays, and we just published the paper. And I asked that you go and read this paper. This is really very interesting because it's not just they have a long axon, they branch arborize. How about function? Are they functional? Do they, incorporate and improve their function? So now we are actually developing this high density multi-electrode arrays to be able to look at the functional connectivity of these neurons, not just axon length and branching arborization. And working together with Dr. Silverman was a blessing because he is one of the best medicinal chemists, and he is the inventor of Lyrica, and he was working on small molecules that improve misfolded SOD1 aggregation. And he identified three compounds, three classes of compounds. And at the time, I was developing this GFP neurons, and we met one of the ALS meetings. And I said, we are actually working on this, and we started collaborating. And we realized that the compound that he identified in his lab, and we call it NU9, does something remarkable. It reduces misfolded SOD1 aggregates, which he identified in using PC12 cells, but we identified that it can also do this in the dissociated upper motor neuron cultures that I showed you. Then we have gone in vivo and used mouse line, SOD1 mouse, and we realized by giving NU9 two doses, 20 milligram per kilogram or 100 milligram per kilogram, there was actually reduction of misfolded SOD1 in the upper motor neurons. And that their mitochondria were so much improved that they were almost similar to wild type levels. And normally in the disease, endoplasmic reticulum is falling apart, and by giving NU9, the endoplasmic reticulum is just like sharp, and it's the textbook structure, like it's beautiful. We said, wow, this is what's happening with just 60-day treatment. And there was less cell loss. Cells decided not to die after you give NU9. They say, you know what? My mitochondria is fixed. I don't have garbage problem. All the protein aggregates are cleaned, and I'm making ATP, I'm making energy, I'm making enough protein. Like, why do I die? Like, why must I die? So this is actually a very good finding, because when we solve the problems of the neurons, why would they die? When we make them happy, why would they die? There's no reason for them to die. So then that's why making these neurons happy is the way forward. Then we said, okay, now we can make the upper motor neurons that are diseased with SOD1 happy. But can we also make upper motor neurons with TDP pathology happy? Then we repeated the same experiment with the TDP mouse, and even though this is different protein aggregation, even though this is different mechanism of action, even though this is a different representation, different underlying cause, yes, indeed, we were able to improve the mitochondria, we were able to improve the endoplasmic reticulum, and again, the cell bodies were still intact in the motor cortex. We did not see cell loss, and apical dendrites were much better. So there was clear, very significant improvement at a cellular level. Wow, this was very nice. Then we said, would NU9 work by itself, or if we combine it with riluzole, edaravone, would they have any additive effect? Then we did in vitro cultures that we added NU9 together with riluzole, together with adarabonin, and we found something remarkable. That when you add NU9 together with riluzole, it's actually added. They're not competing. And then with riluzole, they're not competing. So then, if you think about it, why would they compete? Because they are acting on different mechanisms, right? So NU9, or let's say, for example, riluzole, right, it is acting on excitotoxicity, basically acting on astrocytes, making the astrocytes less toxic and improve the upper motor neuron health. But excitotoxicity is not one of the mechanisms for NU9. So then adding them together could be a beneficial effect. And also, edaravone is free radical scavenger, and that's not really the mechanism for NU9. So then, see, I was telling you mechanism. So if we understand the mechanism, if we understand the underlying causes, then you may actually have different drugs with different mechanisms, and it may be added value, in some patients. And that's why, after we got these results, Dr. Silverman formed a company with his son. It's Akava Therapeutics. And I'm the head of the scientific advisory board for Akava Therapeutics. And we wrote the IND application to FDA to start phase 1. And people told us, don't get your hopes high. Maybe within, 6-8 months, you're going to get rejection. We got approval within 2 months, 2-3 months. I mean, that was remarkable. But then we said, okay, let's try. Let's start. Oh, who knew that clinical trials are millions and millions of dollars? So now, we need to have the clinical money for the clinical trials. But in the meantime, we also need to understand what is the mechanism of action? Where does it bind to? And is it, all these studies. So the good news is that Dr. Silverman actually published a very recent paper with Dr. Klein, and look at that. NU9, you remember I told you, it reduces misfolded SOD1, it reduces TDP pathology, and now look at this. It actually has an impact on amyloid beta oligomers and their accumulations, and that's Alzheimer's disease too. Oh my God. But if you think about it, protein aggregation is a common problem among neurodegenerative diseases. Even though different proteins aggregate, the protein aggregation as a problem is common. And if NU9 can reduce the aggregation of those 3 independent, unrelated, so far apart proteins, how valuable it is going to be for the field of neurodegeneration. And if it works together with riluzole, together with adarabonin, it may not be a drug of its own. It may be a combination drug. Like we may actually take it to reduce protein aggregation and to improve mitochondria, to improve ER. And then for more specific effects, we can take maybe other drugs. So now we wanted to investigate whether NU9 would also have an impact on Alzheimer's disease, on ALS FTD, on FTD. That's why we wrote a grant to National Institute of Aging, and we recently received $7.3 million grant into further investigating the mechanism of action of NU9. And now that, to our knowledge, this is the only compound that can have an impact on those 3 different protein aggregations, how does it do that? How does it do that? So, we are doing proteomics, metabolomics, sets of studies, thermal shift assays to be able to understand where does it bind to. Because apparently that question, where does it bind to, is an immensely important VC question. Every person who wants to invest in a drug wants to know, where does it bind to? But the thing is, for complex problems like ALS and for, these gifted compounds like NU9, they may not be binding to one thing. They may actually be modulating multiple things at the same time or sequentially, or they may have a big impact on maybe a signaling cascade of events. That's why it is taking us a little bit longer to understand how it does what it does. What we know that improves protein aggregation, it improves mitochondria, it improves ER, and it improves cytoarchitecture, especially at the site of apical dendrites. That's why we are very hopeful that, and it's not toxic. So we have the toxicity data, and maybe I shouldn't say it's not toxic, but the toxicity data is in favor of NU9, and I think that's why we received the FDA approval to go into phase 1. So I think once we receive the funding and once we receive the financial support, it's going to be a game changer for neurodegenerative diseases. I am not a businessman. My father was a businessman, and he wanted me to run his company. I did not want to run his company because I'm more of a, curiosity, science, humanity. I am not, I'm not a money person. So, but, I'm sure there are many money people out there, and this is actually a very important protein that we think. So, but let's say you have the protein, all right, fine, and you have the clinical trial, all right, fine, it's not toxic. Now you're going into phase 2. How do you know that it is going to work, right? And I'm going to tell you a biomarker story very fast. Do I have time? Can I go like 5 more minutes? Because
Absolutely.
this is
Yep, you can go. I think there's plenty of questions, and go ahead, and we'll get
all
into
right. So this is very important. I want to, and this is unpublished. So, and I won't be able to reveal the names of the proteins, but I think the study is very important. And here we have a family of ALS patients with SOD1 D90A mutation. Okay. And it is the father. His ALS-FRS score is 39, so he's showing symptoms. He has a son and a daughter. And son and daughter, they both have this mutation. And at the first time, they both were non-symptomatic. But a year later, the son becomes symptomatic, but the daughter remains non-symptomatic. Okay? And here we have another person out of the family, but the same mutation, and his condition is worse than the father. So now you can imagine we have daughters time 1, non-symptomatic, time 2, non-symptomatic, son symptomatic, father, and this. Like we have 5 time points, right? And same mutation. So we isolate serum from those patients, and we isolate exosomes from those serum, and we do proteomics to the proteins that are isolated in the exosomes. Can you imagine? Can you just imagine? Exosomes are like tiny, itsy bitsy, nanometer size. From those exosomes, there is protein. We isolate protein and we do proteomics on that. And then we do comparison analysis between the son and the father and the daughter, and then we also do linear regression analysis, and we use ingenuity pathway analysis to understand what's going wrong. Okay, I'm going to go very fast. First, we compare what is happening in the son, because he was non-symptomatic, now he became symptomatic. So there must be something changing in him, right? And yes, look, the TRIM21 signaling pathway, IL production, activation of NFAT and the immune system, LXR activation, acute phase response signaling, these are all immune reaction related events, and especially TRIM21 tells us the immune reaction also occurs because of the mitochondrial dysfunction. So in the son, when he was non-symptomatic and he's going to symptomatic, he has this activation of the immune response, right? But in the daughter, goes from T1 to T2. For clinical purposes, she's healthy. She's still healthy. She doesn't have any clinical symptoms. She's healthy. But at a molecular level, look what's happening. Vascular problems in the periphery, vascular problems in the brain. Her blood-brain barrier is leaky. That is her major problem at this stage. Her blood-brain barrier breaking up, but she's still healthy. But her blood-brain barrier is breaking up. And she has more post-translational protein phosphorylation. And you remember TDP phosphorylation, accumulation of proteins that are phosphorylated. And she has more protein phosphorylation. She's trying to repair her DNA because she has activated DNA activation. She's trying to overcome some of the problems that she has, and she needs to make sure that her DNA is checked, it's repaired, it's maintained, that, there's no problem with DNA. Her mechanism is very active, even though she's non-symptomatic, she's not diseased, but at a molecular level, she is on fire. And this paper just came up that endothelial TDP depletion disrupts core blood-brain barrier pathways in neurodegeneration. Who would have thought that, right? So then this blood-brain barrier leakage is actually a very, very, very early event. And then if you compare father versus son, right? Because father is ahead of the disease and son just became symptomatic. Look at this. Phagocytosis, complement cascade, B cell receptor, communication this. Oh my God, it's even worse, getting worse of the neuroimmune modulation. So then we said, are there any potential pharmacokinetic biomarkers? And we find couple. Because if you look, son, he just became symptomatic, okay? Father, he's symptomatic, but patient number 2 is more symptomatic. And look, the values are increasing. And if you plot them based on their ALS-FRS score, look at the R-square value. That looks the linear regression, 0.9. Oh my God. And it's not just that protein, it's even the isoforms of that protein, and it tells us where to look for the antibodies and which domain to look for the antibodies. So we find one potential pharmacokinetic biomarker, and I forgot to tell you that this mutation, D90A, is actually affecting mostly the upper motor neurons in ALS patients. And now we found another gene. So again, with even the isoforms, you can see a very nice correlation with the ALS-FRS score with disease progression, that we can actually have a pharmacokinetic biomarker. This is exactly what we needed moving into clinical trials, because now the goal would be to reduce this, or, we know that this is what happens with disease progression. Can we reduce it with the treatment, right? And then how about prognostic biomarker, which means, the daughter, even though she was non-symptomatic, she showed some increase, but the son is higher, father is higher, patient is even higher. So maybe this is the sweet spot. Maybe she was non-symptomatic. Maybe at this spot, it will be symptomatic. Maybe if you look at every 3 months, every 6 months, maybe having that protein in the serum would be a good indication of the timing and extent of when she would be developing this disease. And especially because this is SOD1 mutation and there's tofersen, and then, there are patients who cannot take it because they are clinically okay, like this daughter in this case, maybe we can develop prognostic biomarkers so that they can begin to begin their treatments even earlier. So then I think that I was able to convey my opinion that the brain is important, okay? And the cortical component of the motor neuron circuitry is primarily involved in ALS. And we need to include the brain into the picture of drug discovery. Translation is at the cellular level. You make the cells, you make the neurons happy, the circuitry will be functional. When the circuitry is functional, then the patients will be able to move their arms again, move their legs again, because the circuitry becomes functional. And that's why to make the circuitry functional, we need to make the neurons happy. And we have to pay attention to the common cellular problems. For example, the protein aggregation is a common cellular problem. And if NU9 can overcome this protein aggregation problem at many different diseases, I think it deserves some attention. And it is mechanism. Once we understand the underlying cause of the disease, the mechanism, then we will bring effective treatments to a subpopulation, and before you know it will be everyone. But we can't expect one drug to cure everyone because it's very complex, very different. And I think going from subpopulation to subpopulation is a better way forward. And I think it may be possible to identify disease before symptom onset, like the prognostic biomarkers. And it may even be possible to track disease progression by these potential biomarkers. And I just mentioned one, okay? But there are imaging biomarkers, there are other biomarkers, but they are up and coming. And I'm very optimistic about this, that we would be able to track the disease progression. And that's why I think the future is much brighter, because we will be identifying novel compounds that improve the health of our motor neurons that degenerate due to different underlying causes. And our lab, we are willing to work with other companies and help you identify if your compound of interest also improves the health of the upper motor neurons. We can help you identify that, and we can save you so much money, because if you have, let's say, 5 of your favorite compounds, maybe 1 or 2 of them also impactful in the motor cortex, right? So then knowing that is going to save you so much time, so much money, and it's going to help the field. And I think we can identify novel pharmacokinetic and prognostic biomarkers using, again, proteomic, lipidomic, metabolomic approaches. And we can develop personalized medicine approaches, because every patient is different. And then we can begin treating patients even prior to symptom onset. This is a possibility, I think. And let's not forget the brain. And I would like to thank everyone in my lab, my collaborators. And, it's a huge team effort. And I want to mention Mukesh Gautam. He worked on the new biomarker paper that we have. He will be the first author. He's also worked on the TDP mouse model. And Chris Quintanilla, he worked on the electrophysiology, and he's working on the drug discovery project as well. And we have collaborators from Europe, from drug companies, everyone. And I am thankful to SFN, because look, they highlighted one of our upper motor neurons in the SFN 2024 meeting here in Chicago. So, seeing our GFP neurons up in the wall was a treat. And I am very thankful to everyone who supported our research, foundations, NIH, NIA, Les Turner ALS Foundation, ALS Association, Along Swim, Brain Research, Spastic Paraplegia Foundation. This is a team effort. You cannot do this alone, and we definitely need financial support. Everything is expensive. Everything is required, and the support that is given to our lab is very much appreciated. And I also thank Department of Defense for giving us grants. And I thank you for your time, for your attention. You can reach out to me, my email, my Twitter, Facebook, and our lab information is also here. And thank you so much. I will be happy to answer your questions.
Dr. Ozdinler, thank you so much. That was an incredible presentation about, you're right, about a topic that often gets overlooked. So it was great to have such a focused and robust presentation on the topic of motor neurons in the brain. We do have some questions, so I'm going to jump right in. I know that we're at the a little bit time constrained now, but we will get through them. So first and foremost, why aren't the sensory neurons impacted, since they share the spinal cord and yet they're not involved?
So it's a very interesting question, because again, we need to understand how neurons are born and differentiate and integrate and so forth. So the spinal motor neuron the transcript, if you look at the gene expression profiles of sensory neurons and and the motor neurons, they are very different. And there are also diseases where sensory neurons are affected and not motor neurons. So we can ask the same question, well, in those diseases, why motor neurons are not affected, right? So I don't think that we should expect that when there's a disease, all neurons are affected at the same time. That's why we have selective vulnerability. And in ALS, in motor neuron diseases, it's mainly the motor neuron circuitry that degenerates. But as the disease progresses, it could be possible that, some of this sensory neurons are also affected, but mainly the neurons that are affected are the motor neuron circuitry neurons.
Are there relationships, or what are the relationships between upper motor neurons and non-motor neurons in this sense?
Yes, that's a very good question. So there are many non-motor neurons or non-CSMN in the motor column, right? For example, one of them is callosal projection neurons. So these callosal projection neurons are very interesting because they're also excitatory neurons. They're also pyramidal neurons, and they're not as big or as large as the corticospinal, but they are very similar. And they are born at the same time or similar times, and they migrate together, they reside together. But the spinal motor neurons, callosal projection neurons, you can see them in layer 1, 2, in other layers, but corticospinal motor neurons are here. So, in ALS, the callosal projection neurons remain relatively healthy, but corticospinal motor neurons feel the burden. Why is that? And it may be their projection field because callosal projection neurons project to the other side of the cortex, but corticospinal motor neurons go to the spinal cord. It could be that their gene expression profiles are different. They make different proteins. And, we actually FACS purified these neurons in the ALS mouse and did RNA-seq and proteomics to be able to understand what is the selective vulnerability. And these neurons are totally different neuron populations. And in diseases like ALS, the callosal projection neurons are not affected as much.
Thank you.
Okay, so the next question is there any update on the development and experimental testing of NU9? I know you talked about when the human trials will begin, but just any more information that you could provide.
Yeah. So now we are investigating ALS models, in addition to ALS, ALS FTD, and Alzheimer's disease models. And I actually received funding from CureSPG Foundation, and we collaborated with Dr. Bass on the spastin model. So we're trying to understand whether it would be also useful within the context of HSP. So now, we are trying different preclinical models of different diseases and try to see if mechanistically, that compound would help improve the underlying cause in those diseases that are related and they share common problems.
Thank you. I've got a question that actually speaks to that family history tracking that you showed and the individual progression. So in this case, it has to do with ALS and other neurodegenerative diseases. The question essentially is, my mother currently has ALS and my aunt has dementia. My mother's uncles died of Alzheimer's, Parkinson's, and dementia. I was diagnosed with ALS too. So my question is, I did the SOD1 test, the SOD1 test, but the results have not come back yet. If I were to start using Qalsody because I don't want to waste time, and if SOD1 test results are negative in the future, might this drug actually potentially harm me since I do not have a genetic inheritance?
Okay, so, I'm not a clinician, so I cannot really give advice on, take this drug, do this, they should really talk to their doctors. And as far as I know, the tofersen drug is an ASO, like antisense oligonucleotide, so that it silences the mutation. So if they don't have the mutation, there's nothing to silence. So if they don't really have the genetic mutation that causes the disease, the ASO may not work for them. I think this treatment is basically for patients who have the mutation so that this ASO treatment silences it.
Yeah. And given that it's an ASO, yeah, it would be a precondition, especially given the type of treatment. What is the potential for tofersen to stabilize the disease and even potentially reverse? Has there been any record of reversals or the undoing of the effects of ALS with it?
The question is about tofersen?
Yeah, specifically if you're aware of any of the outcomes so far in terms of disease stabilization or even potential reversal of the impact
I wouldn't be the best person to answer that. I don't know, because I think tofersen has been approved with 60 days of clinical trials, and but I don't know what the follow. I am not the best person to answer that.
Thank you. Celia.
The next question is, what do you think about a prion-like transmission?
Very interesting. Prion-like transmission is actually very interesting, especially there are some papers showing that there may be transmission like that within the context of proteins that are mutated. So let me try to explain what that means. Not everyone may know the prion transmission. Let's say if the protein has one mutation, it makes it a kink, right? And once that structure of the protein changes, when you have a wild type and now they interact, and then boom, this also becomes like mutated, even though there's no mutation and it's just wild type protein, because one of them has that structural change. It starts a haywire and makes every protein different. So I think this was like a big theory at the time, that SOD1 mutation, for example, may have a prion-like progression, or some kinds of TDP pathologies and so forth. It may be possible, but I don't know to what extent it could be possible. Like theoretically, it may be possible. And I think the location of the mutation is also very important, because not all mutations are the same. And within the SOD1 gene, I mean, how many mutations have we detected? Was it 89, 90, 120? Like it's not just one mutation, right? Multiple mutations. Same is true for TDP. And the location of the mutation is actually very important. And we cannot say, oh, mutated protein does that. It all depends where the mutation is.
Thank you. There was a question about using sensor arrays. So how would you place the sensor array in a brain-computer interface? Would more than one site be better?
Sensor array. Can you repeat the question again?
How would you place a sensor array in a brain-computer interface potentially? And would more than one site be better?
Okay, so this is an interesting question, because for some cases now they are testing having sensors either put in let's say in the motor cortex, to be able to modulate or excite or read or transmit brain's activity, and together with another edit machine or reader. And one of the problems with the ALS, remember I told you that the apical dendrites degenerate, right? And if the apical dendrite degeneration is so massive, and that you don't really have those apical dendrites where they have all the input, right? And if you put in the surface, but the cell body is in the deep layers, and you put it in the surface, it would be harder for them to hear each other or listen to each other. Like you really need healthy apical dendrites. And I think then, modulation early in the disease would be more effective than later in the disease. But I think that this brain-machine interface is a very, very important topic, and it is developing really fast. And some of those are also biodegradable, and some of them may be transplanted. And I know that there were studies with different large mammals. Like I think that they have done studies in primates and so forth, either like in the brain or like putting vertical into the motor cortex, right? So I think those experiments are still ongoing, but I'm very hopeful about their success.
Thank you.
The next question is, at present, is there a way to determine the underlying cause of the disease of ALS?
Okay, so this is actually a very, very important question for me, because that's what I would like to do. I would like to work with every patient and get their serum and plasma and do the proteomic, lipidomic, RNA-seq, and metabolomic, and use all these, ingenuity pathway analysis or large data management toolboxes, put all the data together and begin to understand what is the upstream regulator, what is the converging path, what is the canonical pathway, what is the main problem, so that, the patient actually carries that information with them. The information is in their serum, is in their plasma, is in their CSF, sometimes in the urine, but all the biofluids, they have that information. But we didn't have the capacity to understand what that information is. Because you can have data, But then from data, you must generate knowledge. And from that knowledge, you must also generate understanding, right? So, like for example, if I tell you you have gene A, B, C, up, down, like, okay, what does it mean? But then if this gene A, B, C, D are working together on the axon transport pathway, oh my God, like you have three genes that are upregulated, downregulated, but they're all in the axon transport pathway. So then you may think, all right, so then maybe the axon transport pathway is the most affected. Do you see? So I think that we need to develop personalized medicine approaches. And I really want to form a company. And I even have a name for it in my mind. And, I would like to help each and every patient try to understand what's really going wrong with them. And that information, I think, is going to help their doctors to identify, which clinical trial would be best suited for them. And also, if there are any drugs out there that are FDA approved for other diseases, other conditions, that may help their specific condition.
Thank you. There are, you gave great examples of the fact that the more we know about the disease, the more we know about root cause of the disease, the more we're likely to move into this space of population segments and subpopulations. And that triggered a question from the audience that I think is right on, which is oncology routinely uses multiple compounds to treat all the different types of cancers. How about combining NU9 and riluzole and an immunomodulator like Koya? Is that something that you envision happening with the disease as we get to know more of it?
Yes. And you know what? The cancer field is about 10 years ahead of neuroscience field. So the cancer, they know about this selectivity and different tumors, like cell type specificity, different like cancer field. When they are like our big brothers. They have solved many of the problems that we are still struggling. And I think in the future, this is exactly right on. Like, I think the more, like, to be honest with you, okay, when you think about FDA and drug discovery and, applying to FDA for drugs and so forth, you have to tell them the disease name. You have to say that this drug is for Alzheimer's, this drug is for Parkinson's. So you need to tell them the disease name. So FDA, when it was established in the 1940s, at the time, we did not know about RNA-seq. We did not know about proteomics, lipidomics, metabolomics, all the omics. We did not know. And the diseases were characterized based on clinical manifestations and clinical outcomes. So they said, oh, Parkinson's patients does this, Alzheimer's patients does this. And at the time in 1940s, it was expected that each disease is a distinct entity that, and the goal was to cure the disease. The goal was to cure the disease. But now, 2025, we know that Alzheimer's is a little bit of Parkinson's, a little bit of this, and ALS is a bit, because if you look down, they have very similar problems shared, like protein aggregation. You see it everywhere. Mitochondrial problem, you see it everywhere. But in 1940s, they did not know this knowledge. So now, with the knowledge that we have, right? We have all these new dresses, new hats, new this, but we are trying to fit into the old school definitions of, and that's the one of the main problems, because we need maybe clinical trials that say, all right, here's a clinical trial that improves the health and integrity of mitochondria. This is the best mitochondrial drug, whatever. And then to that clinical trial, we are accepting ALS patients who develop the disease mainly due to mitochondrial dysfunction. We are accepting patients, Alzheimer's disease patients, Parkinson's disease, blah, blah, blah. So then, it's not really the disease that we are curing. It is the mechanism that went wrong in these patients, and we are overcoming the burdens of the mechanistic dysfunction. I think if we change our mindset, oh my God, like it's going to be a game changer, because currently we're trying to cure ALS, but ALS is so heterogeneous, and that, one drug So it's like we are waiting for Godot, because the Godot is going to come, one drug cure everyone. It's not going to come. One drug is not going to be good for everyone because everyone is like apples and oranges and grapes and this and that. It's just so much. So that's why for every clinical trial, I think that's why we have a subpopulation that benefits, maybe 10 people, 20 people, but we currently ignore them. We say, no, it's not significant. It's not significant. But for those 10 people, it's significant. Those 10 people, they matter, and I think it's because of our expectation that one drug is going to cure everybody or is going to be the result for every problem.
It's a great point. It's one of the reasons we do the work here at Everything ALS to do all these studies with the sensors to really get to those subpopulations so that upfront clinical trial designers have clearer subpopulations to work with to target their studies and hopefully change that signal to noise ratio that seems to be holding back the results of so many different trials.
Definitely.
Thank you.
Definitely.
Sylvia, you had the next question?
It's a follow-up on NU9. Have you been able to determine if NU9 has any effect on neurofilaments or whether we talked about it, if it's clinically significant?
Yeah, so for the neurofilaments, how you mean change the levels of neurofilament?
Correct.
Okay. So we have actually given NU9 to SOD1 mice and TDP mice, and then after giving this treatment for 60 days, we isolated their brain, we did the proteomics, and then we also collected their plasma from the blood and then did the proteomics to be able to see what is changing over time and with treatment, okay? So in addition to neurofilaments, there are many other proteins that come up. So yeah, neurofilaments, I think that, it's very important, but especially within the context of upper motor neurons, I think that there may be other proteins that come up as well.
Thank you. All right. So in terms of what we've covered tonight, can this protein help with upper motor neurons that already have been damaged? Is there reversal that's possible? It sounded like from at least the cellular environment, you're seeing some of that.
Yeah. So, we started our treatment at P60. And P60, postnatal day 60, is a time when these mouse models begin to show symptoms. And we know at a cellular level that at P60, a significant amount of corticospinal motor neuron already degenerated. So there's ongoing degeneration already. Okay. But then so you may think death happens overnight, but actually death is a process. So, every day we are actually dying one more day, like, it is a process. And neurons, which are vulnerable to degeneration, they feel the burden, it's a process. But once you solve their problems, okay, during that process, then you either slow the process or they stop and say, well, these were my problems and they are solved. Okay. And why was I supposed to be upset again? And then they may not choose the path for neurodegeneration. They may actually choose the path for neuronal integration. And that's what we have seen. So with 60-day treatment, even though there was ongoing neurodegeneration, once you solve their problems, they may actually integrate. So I think I'm very optimistic that especially if we start treatment early when they are vulnerable, but they are not dead yet. Like, they are still hanging on. They're still hanging on. We solve their problem. They may respond. They may respond and say, you know what? I think I can do. And then they get the information, and then they send the information. I think that we can rescue them. But if astrocytes have eaten them, microglia has eaten them, and there's nothing left, then it's hard. But this nothing left stage happens at the very, very, very end. So I think that most of the patients would still have neurons to survive.
We keep fighting.
Yes.
Um,
And you know what? I want to tell you this because, I became very good friends with those upper motor neurons and they speak to me now when I do the RNA-seq and proteomics and so forth. So when we do RNA-seq to them at P15, P30, P60, P90, P129 different times, you would be amazed how resilient they are. Even 120, like this is the end stage, they call it. It's the end stage. No, 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 no. Even at the end stage, they express some of the growth factors. It says, I'm here, like I'm just holding. Even to the last day, they're holding on. And that gives me like goosebumps. And they're holding on even to the last day. Like, who am I to say, oh, who cares about the Bermuda? We when they are really hanging on and when they are really doing the fight at a cellular level, they're still there. I think we make them happy. They would say thank you, and then they will say, all right, where was I? Let me build this. Let me build that. Let me do this, and because they know how to do it, and I think that we can really help them.
Thank you. So there's a question from the community who are obviously deeply motivated here. What can we do to help you in your research?
Oh my God. Oh, I think community building is really important, and attending meetings like this is really important. And I don't know, maybe we can establish fellowships. Fellowships are very important because then it helps me to train the young generation of scientists and maybe introduce me to the key philanthropists or drug companies who are interested in this. Be the enzyme, be my friend. It's not just only money, it's also the human touch. It's also, we are all in this together feeling. And I'm writing grants all the time, and I don't know which grants is going to be funded. I don't know. It's very depressing right now, and we just have to push together to move the field forward.
Thank you.
I think a good follow-up question for that is, have you had any discussions with Niels about collaborating somehow at all, or any other big organizations within the community?
So we are part of Niels. I'm part of Niels, and also there's an upper motor neuron group at Niels, and I was the co-chair last year at the Niels meeting. So I'm very much connected with my friends at Niels, yes.
Thank you. Anything we should be eating to help our brains and to help feed these beautiful forests of sequoia-like motor neurons that you've shown?
Yeah, so to be honest with you, I was diagnosed with sleep apnea recently and severe sleep apnea. And I started reading into sleep apnea, and I realized that problems with sleep is actually very common in patients with neurodegenerative diseases. So maybe to help ourselves, we really need to have a very good sleep. Because the brain is in CSF. The brain is swimming in this CSF, the liquid. But the brain, as it's working, it has all this toxic things and dirt and blah blah blah, and it secretes to the CSF. So then the brain is swimming in this dirty water, and this dirty water cleans when we sleep. Okay, and that's the only time when it cleans. And we have about 850 mLs of CSF. And when we sleep and our heartbeat becomes like, regular, then this CSF is now being cleaned. But if we don't sleep well, if we have sleep apnea, then this CSF is not cleared. It's not cleaned. And imagine for 9 years, for 10 years, for 5 years, that CSF is not cleaned. You know what happens? Protein accumulation. And all these sporadic cases of neurodegeneration, no mutation. It may be that the CSF, where the brain is swimming in, is dirty. So I think to keep our brain healthy, we have to sleep well. And I did not know this, or I did not appreciate the importance of this before I was diagnosed with sleep apnea. So that's why I ask every patient to ask themselves or to check if they have sleep apnea and how is their sleeping. Because if you sleep well, if you clean, the liquid that surrounds your brain will take a deep breath and your brain will be better functioning. And again, oxygen levels must be high. And, doing exercise would definitely help. But I think sleep is very important.
Thank you.
Thank you. We have one more last question. It's a two-part question. There was an idea that was put forward that even in a patient with complete loss of function, not all motor neurons are lost and the remaining ones that can take over the function of others. This was based on a few patients who had recovered on their own. So it looks like it's got to do with something with brain neuroplasticity. So taking all of this into account, do you think AKV9 could have an effect on a patient who's completely lost their functions? And based on what you've seen with experiments, and then have you investigated neurofilament biomarker improvements with AKV9?
Yeah. So to answer the first question, we must do the experiment such that, we start treatment not at P60, but let's say P90, or, at a different time point, And, how late can we go to see the effect, right? We haven't done those studies. So, I wouldn't be able to give a good and truthful answer. So I don't know. I don't know what is the latest that we can go. And for the neurofilaments, we need to do this in the clinical trials. So, we haven't done the neurofilament testing in patients. I think in phase 2, that's what we're going to look at.
Thank you so much. Christian?
Great. I think that's about it. We are so thankful for your time. I know we went over and you stayed with us throughout. It's a testament to your dedication to the community that you make this kind of time for us, and we're so thankful. I think I will say again, it's so important for the community to know how important it is to participate in research, to participate in the activities that really help us understand the underlying disease and how to segment and get people ready to be able to participate in studies with greater data collection and greater understanding of your individual unique stories. Every single one of you, your data is your life story in many ways. So we hope that you'll participate. And with McFinn, I'll turn that over to you now. And one final thing. This presentation and the Q&A will all be available, as Sylvia mentioned also in the chat, via on demand on YouTube. We will also email it out to the community when it's available. So please don't feel like it's a one and done. If you have additional questions, you can also access it inside the Everything ALS app, where there is a chat kind of experience, a generative AI chat experience, where you can continue to ask questions of this presentation, as well as the many other presentations that are in that library. So by all means, keep in touch with us, and we look forward to seeing you in the next presentation.